All right. Well, good morning, everyone, and happy Friday. Thanks so much for joining us today for the COVID-19 Virtual Research Seminar Series. My name is Melissa Rogers. I'm Associate Director of Business Development and Industry Relations at the Mesh Academy at Keck School of Medicine. So our mission at the Mesh Academy is to empower the convergence of research disciplines to address the challenges in human health and disease. And we're very excited to be able to bring this series to you all. Oops. All right, so today we have 3 speakers from Keck School of Medicine, and they're gonna be telling us a little bit about some of their COVID-19 related research. So we have 3 presentations, and we are gonna do a Q&A session following the presentations. So if you have any questions, please use the raise your hand feature. And when we get to the Q&A session, we'll be able to unmute you and you will be allowed to ask your question directly to the speaker. We are also recording the webinar, so it'll be available later for those who aren't able to stay for the whole session. So we will get to our speakers now. So we have a great program today. It's my pleasure to introduce our speakers. So our first presenter is Dr. Weiming Yuan. He is Associate Professor of Molecular Microbiology and Immunology. And today he is going to be telling us about suppression of the human innate immune system by SARS-CoV-2 virus. So, all right.
Okay. Okay, can you hear me?
Yes.
Okay, thank you. Does this work? Can you see my slides?
Yes, we can see it. You might want to switch it to just the presentation mode. It's in presenter mode right now.
Sure. Okay. All right. Good morning. Thank you, Melissa and Mary and the MASH Institute for inviting me to present my work. I have to say that by now, my work is mostly a work in progress. So, I try to give a presentation of the field that give you highlights of the best studied mechanism within this innate immunity suppression by SARS virus. So it's mostly the published work in the literature. So I'm giving presentation about the suppression of innate immunity. So what we want to do is to first go to the basics and just to remind everybody the immune process, immune response for any infection. So this is a textbook figure from January Immunobiology, just to remind everybody. So we have for any infection, we have an immune system for first innate immune response, and as you can see here, this is the y-axis is the level of the microorganism, either virus or bacteria. It's a similar overall, but for any infection to start to be a productive infection to cause disease, the microorganism has to suppress the innate immune system to break the immune system so that the infection can start. Then the level of microorganism increase, and to a certain level, it was initiated the adaptive immune response. And once the adaptive immune response initiated, the level of the microorganism will quickly go down, and eventually, if it's active infection, the pathogen will be cleared, and then for adaptive immunity, you have memory. So my work mostly focus on the innate part. So I want to review the innate immunity system. Then it's composed of physical barriers and also internal defense. So physical barriers including skin and the mucosal surface and a digestive enzyme in the mouth or the stomach. All these are part of the innate immune system. From the immune system side, there are inflammatory response, there are complement proteins, there are phagocytic cells and other immune cells. I want to say a little more about this immune components. So for the cellular part, we have even stromal cells for NSA. For SARS virus, we have the long epithelial cells, and those stromal cells have their innate response within the cells. And we have immune cells, different immune cells, monocytes, macrophage, dendritic cells, neutrophils, eosinophil, or basophil, or mast cells, or NK cells. For the soluble part, we have complement system, and also cytokine and chemokines, lysozyme, or active phase protein. My talk will be focused on mostly the cytokine. So for the SARS virus, the best studied innate immune response suppression is it's a suppression of the type 1 interferon, which is part of the cytokine system. So here I want to first give you an overview of, this is a paper in preprint. It's by Italian group. They summarized several dozens of patients. They correlate the immune response and the virus load and with the disease progress. You can see typically SARS-CoV-2 virus takes 5 days from the initial contraction of the virus for the average 5 days from the contraction of the virus to the first sign of symptom. Okay, then the virus load keeps going up. Then you can see the from the onset of the symptom to for many patients, typically 8 to 9 days, start to see severe symptoms like pneumonia and around the same time you can detect the IgM or IgA or IgG. And if the immune system is balanced well, then most of patients will get mild disease and the virus shade will go down. But for some patients, you will have a more severe pneumonia and complications for even eventually patient died. Okay, as I mentioned, I will focus on the SARS virus suppression of the cytokine, specifically type 1 interferon production. This is the best studied mechanism for SARS virus. And there are so many reports about how the SARS virus, a very similar virus to SARS-CoV-2 virus, how this SARS virus can very efficiently suppress the type 1 interferon. And this is one of the first few reports. There are several reports from China. This report is from Singapore. What they show here is they isolated the PBMC from this, at that time it's 2003, and they collected the PBMC from the patients infected with the SARS virus or patients infected with the seasonal flu or control. What they did was RT-PCR to measure the amount of the different cytokines and also inflammatory proteins. You can see there is very severe inflammation in the SARS-infected patients, PBMC. However, it's very clear that the type 1 interferon alpha and beta, it's almost completely suppressed in the patient infected with SARS virus. So this is the first, one of the first few reports showing the type 1 interferon is suppressed. As you know, the type 1 interferon is very important that for the early hours defense for the patients to fight against the virus, because it's a paracrine cytokine. It actually mobilize the cells to a so-called antiviral state, and then for the mechanistic study, people use the mouse model to see how this type 1 interferon suppression plays a role in the SARS virus pathogenesis. You can see that what they use biopsy infected with the SARS virus, and what you can see is that the type 1 interferon is not detected until 24 hours after infection, which is a very long time for active virus infection. Okay, and and if you supply back the type 1 interferon early on during the infection, so you either supply the type 1 interferon either 6 hours or 12 hours or 24 hours after the infection, you can see the mouse the disease severity is much less. You can see the mouse weight loss. This is the earlier the better. So 6 hours give the best result after the initiation of the virus infection. The mouse quickly recover. The 12 hours is still recover, but it's slower, and this is a survival. If you don't treat, the mice will die. If you treat early, if you treat early, mouse all recover, and the treat later, some recover. And if you treat too late, 24 hours, most mice will die. Okay, so this is well studied in the mouse model. Then we, based on these studies, early on during the SARS COVID-19 pandemic, there are more studies. I just want to quickly mention this, okay? So what kind of virus gene actually suppress this type 1 interferon suppression production? And there are reports that show at least 3 viral genes. It's a SARS virus genome, and it's operating frame 3B, ORF6, and the nucleocapsid protein. At least these 3 proteins would suppress the type 1 interferon production. And based on this study, as I mentioned early on during the pandemic, in a Hong Kong group used the type 1 interferon in their clinical trial. So what they did here was they use type 1. This is either tri-treatment or biotreatment. The biotreatment meaning that they have 2 antivirals, and the tri-treatment is the 2 antiviral medicine plus interferon beta 1. So you can see this is a disease score. The patients, some patients recover, and for the antiviral treatment, the disease score is not as good as the treatment with the interferon beta. And then if you look into the more the virus titer in nasopharyngeal or post oral or pharyngeal or the throat swab, all the virus titers are lower. And you can also see the cytokine, so-called the cytokine storm. It's much less severe in the patients treated with the interferon beta-1. Okay, this is the IL-6 amount. Okay, so what we are working on is the SARS-CoV-2 virus. And this is the CoV-2 genome it's largely similar, but there are a few noticeable difference in addition to the much higher affinity of the spike protein to the ACE2. And there are missing ORF3b, which is one of the interferon type 1 interferon suppressor, and the ORF8 is also different in the genome structure. So and for my work, I work on other innate components of the immune system, because as you can see, treatment with the interferon, type 1 interferon, it's not the whole story. It's not like cure everything, just rescue all the patients, but they're suggesting the virus have other mechanisms targeting different components of the immune system. Innate immune system. I just want to remind you that the innate system have multiple, as I mentioned to you, there are cellular components and soluble components, and there are multiple different innate lymphocytes. There are also so-called innate-like, which is have some feature of adaptive immunity, but is still limited receptor diversity. So including the NKT cells and the mate cells. And for typical adaptive immunity, you have B cells, T cells with very sophisticated VGG recommendation. So we work on mostly this part of the innate immunities, and I just want to emphasize one more point of the innate-like T cells, like iNKT cells and mate cells, and this are innate-like because they have a limited repertoire of their T cell receptor, and I work on on these two groups. Right now, we are trying to identify how the SARS-CoV-2 virus evade this two group of T cells function using expression library, infectious clone, and we're making some progress, but that's not the whole story to report yet. Okay, that's my presentation. So I hope I can report more in the future.
Great, thank you so much. Our next presenter is, let's see. Okay, our next presenter is Dr. Ya-Wen Chen. She's assistant professor of medicine in stem cell biology and regenerative medicine. And today her talk is modeling of human respiratory virus infection in human pluripotent stem cell derived lung models. So please welcome Dr. Chen.
Hey, thank you for the nice introduction. And I want to thank the MESH for inviting me to kind of introduce what we can contribute to the USC community about the SARS-CoV-2. So today I'm going to talk about what kind of models we can provide in the lab for respiratory infectious disease and what have been used for to model using this, the lung models we can provide. So I just want to first start with a little bit lung developmental background. So the lung develops from the primitive gut tube. At the anterior ventral side of the gut tube, the lumbar forms, followed by then it separates from esophagus, followed by a complex branchial morphogenesis. Then the proximal distal specification arises, followed by the formation of alveoli. Oops. So alveolar maturation occurs late during development with the, and continues after birth with the generation of alveolar epithelial type 1 cells, these flat guys, and type 2 cells, these cuboidal guys. Type 1 cells are essential for gas exchange, and type 2 cells produce a molecule called surfactant that is critical for the maintenance of alveolar integrity. Besides the alveolar cells, there are also different types of cells in the airway to help us maintaining the function of the lung, such as club cells, goblet cells, multiciliary cells, and basal cells. So the goal of my lab is to generate these different types of airway and the lung cells using what we call human pluripotent stem cells, including embryonic stem cells and induced pluripotent stem cells. The capacity to do so in vitro could serve as model to study human lung development, drug screening, as well as disease modeling. And an ultimate goal in my lab is to utilize this in vitro generated lung cells to sit down decellularized lung matrix. And hopefully one day we will be able to reconstruct a functional lung and qualify for transplantation back to patients. However, this route still needs decades to go. Lung is a very complex organ. So in the near future, what we would like to do is to use this in vitro generated lung and the airway cells to transplant into patients' injured lung. And hopefully these lung cells could help the patient to repair their lungs and regain their lung function faster. But today, I'm going to focus on what models we can provide to model respiratory infectious disease. So our lab focuses on using human pluripotent stem cell derived lung models. And now we have two main models in the lab at this moment. One is the two-dimensional lung culture model. The other is the three-dimensional lung organoid models. Both models generate majority 82 cells with less than 5% of other lung epithelial cell type. So for the 2D lung models, we were able to detect different types of lung epithelial cell markers in the culture around day 50. However, this 2D culture system does not contain the necessary mesenchymal cells, and they don't have a proper correct structure like we have in the native lung. And the 2D lung cultures have been used to model influenza virus and the impaired interferon amplification in human IRF7 deficiency. So influenza A virus first target the entire respiratory tract with the viral antigen presented in type 1 and type 2 cells in human. So we first generated patient-specific lung cells from iPS derived from IRF7 deficient patients' fibroblast. And the influenza A virus replication and interferon beta induction were compared with two control lines. And the IRF7 expression in response to interferon alpha, interferon beta, and the interferon lambda 1 treatment. And the interferon beta production in response to influenza A virus infection were impaired in patient lines. And the overall titer for influenza A virus appeared equal regardless of the IRF7 phenotype. And it's largely because of the efficient replication in cells that's not lung cells in the 2D culture. However, when we score the infected lung cells for influenza A virus nucleoprotein antigen, we saw around 50% of the patient's lung were double positive and versus 27% in the control. So this data show that we can use the 2D lung models to model influenza A virus infection. Another models that we can provide in the lab is a three-dimensional lung organoid model. In this model, lung organoids were cultured in submerged culture condition first, then get embedded into the matrigel to generate this branching morphogenesis. And by around day 50 to 70, the branching organoids express all sorts of lung epithelial cells markers, with around 5% of the total cells are mesenchyme cells. And this model has been used to model RSV. And RSV infection is the most common cause of acute severe lower airway disease in young children. There is currently no vaccine available. Its treatment is extremely poor. You'll see the kids keep coming back to the clinic with the same infection. There is currently no perfect animal model to mimic RSV human infection phenotype except chimpanzee. A couple papers showed that the RSV can infect CD80 cells, but if you see the patient samples, you can see 81 and 82 cells were also infected. One of the key features for the RSV infection is the sloughing of infected cells into the small airway lumen. So this is most likely the major factors that we see the small airway obstruction in this sick kid. So when we infect the lung organoids with RSV, 1 day post-infection, we didn't see any sloughing of infected cells into the lumen of the organoid. However, 2 days post-infection, we started to see the infected cells kind of detach from the basement membrane and moving towards the luminal space of the lung organoid. So this shows that the organoids can also be used to model infectious disease, RSV. Besides RSV, the lung organoid model was used for para-influenza infection as well. And this is a collaboration with Dr. Moscona's lab at Columbia. And we think to characterize pathogenesis or molecular evolution of the para-influenza virus type 3 fusion complex gene and in the entire genome of para-influenza virus clinical isolates, it is important that the virus be propagated without adaptation to laboratory condition in immortalized cells or providing selective pressure. So here we show that the titer of the para-influenza virus type 3 and the clinical isolates after growing 3, 5, 7, 9 days in the lung organoids. And here you can see the virus spreading in the lung organoid. And the spread of the recombinant GFP expressing clinical isolates through the lung organoids at day 3 and day 9 after infection can be seen. And to determine whether the clinic isolates virus propagated in the lung organoids preserve their genomic sequence or show evidence of selective pressure, we analyzed the whole genomic sequencing of the virus collected during the lung organoid infection and compare them with the sequence of the same virus grown in parallel in either human airway epithelial cells or immortalized monolayer CB1 cells. So here you can see that even 21 days of infection, after 28 days of infection, clinical isolated para-influenza virus type 3 passaged in lung organoid culture showed no genomic changes compared with the CB1 cells culture at day 14. So the viral genomics that's preserved during the growth in the lung organoid, and we think the lung organoid culture is actually a better model to study infectious disease than the traditional 2D models. So in addition to address whether the lung organoid model may serve in the future to compare and the differentiate models of infection between important viruses that infect the respiratory tract, we infect the lung organoids with measles virus as well. So measles virus is known to infect respiratory tissue via basolateral surface in distinction to para-influenza virus type 3. And measles virus infection induces syncytial formation even in healthy hosts. So here we can see the syncytial formation caused by measles virus at 2 days after infection. And this shows that we can also use the lung organoid to model measles virus infection. And this is a recent paper that just got deposited on the bioarchive. So this group actually used our kind of combination of our lung differentiation protocol. So they started with human pluripotent stem cell and culture the lung cells in 2D fashion up to 25 days. And then they embedded these lung progenitors into the matrigel. So they actually kind of form spheroid, not organoids. However, they did show that these lung spheroids using our differentiation protocol, were able to be infected by SARS-CoV-2 pseudovirus. So this is very encouraging. And in my lab right now that we just got a green light approved to go back to the lab, and we are gonna work on 2 different SARS-CoV-2 projects. One is the collaboration with Justin. I think he will mention the project in his talk. The other one is we are trying to target TMPRSS2 as a therapeutic target for the virus. So TMPRSS2 is the protease that the virus uses to prime its spike protein to enter the cells. So we had a panel of around 20 monoclonal antibody generated in the lab. And we also, from the RNA-seq data, we know that in our lung organoids, we have good amount of ACE2 expression as well as TMPRSS2 expression. So we are currently trying to see if treat the cells, the lung cells or lung organoids with these antibodies could block the SARS-CoV-2 entry to the cells. So besides the in vitro model, we have 2 potential in vivo models that we can provide in the lab. So the first one is a kidney capsule transplantation. So what we can do is that we can generate the lung organoids and we can transplant these lung organoids under the kidney capsule of immunodeficient mice. And here you can see that 1.5 months post-transplantation, these organoids grow into this large tubular airway-like structure, light up with pseudo-stratified airway epithelial cells and surrounded by these smooth muscle cells. And 5 months later, you can see they started to generate this branching morphogenesis. 7 months later, we started to have alveolar-like structure with all the lung epithelial cell types that we know that we can find in these outgrowths. However, this model takes a long time though. The second model we can potentially provide, it's another humanized mouse model. So in this model, we injure the immunodeficient mouse lung, and we have another type of progenitor cells generated in the lab. We can intranasally transplant these progenitor cells into the mouse lung. And 3 weeks later, we saw a good human cells reconstituted in the injured lung. And in the airway structure, we see these human cells, the white color, express SOX2, proximal airway marker, NCAS 2.1, general lung cell marker, surfactant protein B, club cell markers in the airway, NGFR basal cell marker, acetyl alpha tubulin, multiciliated cell marker, and mucin 5B goblet cell marker. In the more distal area, the human cells also express both type 1 and type 2 cells. So this is another model that we can potentially provide, but this model is still not very stable at this moment. The kidney capsule one is more stable. So with that, I would like to stop here to thank my previous postdoc mentor, Dr. Hans Schnock and Dr. Moscona and Mattel for the support for the viral study. And Sarah from UT Health started with the lung differentiation protocol, and Dr. Zia Borak and Dr. Rosen for giving me the chance to have a lab at USC. And of course, my beloved people in the lab who pushed me to get money. Thank you.
Thank you very much, Dr. Chen. So for our final presentation, we'll be hearing from Dr. Justin Ichida. Dr. Ichida is John Douglas French Alzheimer's Foundation Associate Professor of Stem Cell Biology and Regenerative Medicine. And today he'll be talking about development of novel PIKFYVE inhibitors for the treatment of COVID-19. Please welcome Dr. Ichida.
Okay. Can you guys hear me? Sorry, I was on mute.
Yes, we can hear you.
Okay, good, thanks. Yeah, thanks for the invitation. I really appreciate the opportunity to talk about this collaboration that is between our lab and Ya-Wen's lab and a startup company called Stem, and it's funded by CERM. The goal is really to identify or develop inhibitors of a kinase called PIKFYVE for the treatment of COVID-19. So you're probably familiar with the infection methods that SARS-CoV-2 can use to enter cells by this point, but just to reiterate, there are two different pathways that it can use to enter cells, either an early pathway or a late pathway. And the early pathway is where it binds to the AC2 receptor, but that TMPRSS2 protease on the plasma membrane can cleave the spike protein on the virus and allow its membrane to fuse with the plasma membrane of the cell and enter the cytosol that way. There's a second pathway that does not require that TMPRSS2 protease. It's just straight endocytosis of the viral particles. And in this way, it can enter cells that don't have that protease. This method of entry, we find, is regulatable or can be blocked by inhibition of PIKFYVE kinase, which is what I'm going to tell you about today. The remdesivir drug, as you know, really interferes with the replication of the RNA genome. And we know that PIKFYVE kinase regulates different aspects of vesicle trafficking. So we wonder if it also regulates these aspects of repackaging the virus or budding of the virus out of the cell for spreading the virus. But what we do know right now is that it's really working on this early entry step, and that's why we're trying to develop an effective drug to it. So we were flagged by this potential indication because some of our colleagues down at Sanford Burnham, Sumit Chanda, and some people from the Scripps Research Facility did a large screen on to look for known chemicals that could inhibit SARS-CoV-2 infection. And they screened 12,000 compounds, which includes essentially all FDA-approved drugs, and almost all drugs, if not all small molecules that have ever been put into humans. And so out of all of those 12,000 chemicals, this PIKFYVE kinase inhibitor, apilimod, was the most potent at blocking infection of live SARS-CoV-2 into cells. And you can see that it was about, it's about 25-fold more potent than remdesivir. And so this really told us that, this might be a good target to develop an inhibitor for. And this is why it blocks that late mode of entry into the cell. So what happens is these viruses, and many of these different viruses will be endocytosed into the cell. They'll be basically stuck in an early endosome. That early endosome has to mature and transition over different types of endosomes into a late endosome, and then fuse with lysosomes for the SARS-CoV-2 virus to be released into the host cytoplasm. And the reason is because in the lysosome, there are cathepsins, specific cathepsins, that also can do something similar to what TMPRSS2 can do and cleave the spike protein on the virus, which allows that viral membrane to fuse with the lysosomal membrane and eject the viral particles into the cytosol. So PIKFYVE is required to mature early endosomes into late endosomes where they can fuse with lysosomes. And so by blocking that PIKFYVE activity, you actually change the membrane composition of phospholipids on these early endosomes, which keeps these early endosomes in the early endosome state and prevents lysosomal fusion, and therefore prevents the viruses from escaping into the host cell cytoplasm. The second thing that PIK5 inhibition does that could be very helpful for COVID-19 is that it's known to temper the Th1 immune response, which is a key part of the cytokine storm that occurs and leads to the deadliness of the virus. So you can see in this cartoon, essentially there's a hyperinflammation, including IL-6, TNF alpha, and other cytokines. Part of this is an important part of this cytokine storm includes interferon gamma, and interferon gamma is excreted in part through this feed-forward loop between interferon gamma and IL-12 signaling that goes back and forth between T cells and macrophages. And so if you block one link of that, you can significantly temper the amount of interferon gamma that will be produced. And so you can see here that PIK5 inhibition has been shown in several studies to be able to temper this or lower this feed-forward response between IL-12 and interferon gamma signaling, which leads to a lower overall amount of IL-12 and interferon gamma. And so we think that aside from its ability to block infection, its ability to temper the cytokine storm may be an equally important aspect of potential therapeutic efficacy. So the reason why we even know or are working on PIK5 was because we had originally done, our lab generally studies neurodegeneration, and we've been trying to find therapeutic targets for ALS. And we did a screen where we used ALS patient nerve cells in a dish and screened several thousand compounds for the ability to rescue the survival of these ALS patient neurons in vitro. And PIK5 inhibitors were the most potent class of compounds that could do this. You can see here, this is the ALS neuron survival with just a vehicle control, and this is with treatment of a PIK5 inhibitor. This is a curve kind of describing similar results where you have the survival of patient neurons here. They degenerate very quickly in a petri dish, but if you add the PIK5 inhibitor, it increases their survival. If you change the structure of that PIK5 inhibitor so it has poor PIK5 inhibition activity, a lot of that rescue activity goes away. And this is very specific for patient cells because control neurons are not affected in their survival towards PIK5 inhibition. And I think that also speaks to the, any potential neurotoxicity that you might worry about with PIK5 inhibition. There's really no change in the survival of healthy neurons with PIK5 inhibition here. So the problem with apilimod, which is the best known PIK5 inhibitor and the one that was identified in the screen from Scripps and Sanford Burnham, the problem is that it's really not suitable for in vivo use. And the reason is because it's highly susceptible to liver metabolism. So this is a PK study done by Akirstem on apilimod, and you can see that after oral dosing, it falls quickly below the concentration needed to effectively inhibit PIK5 in the plasma. So really, within 2 hours, it's not going to be effective. What Akirstem has done is medicinal chemistry to identify new inhibitors of PIK5 that have a much longer half-life in vivo. So you can see here this new red molecule, ASR149, can be above that concentration for at least 8 hours at the 50 mg per kg dose. So what they've shown is that these new structures, such as ASR149, are not only better at have more stability in vivo, but they also retain the same potency. So this is a purified enzymatic biochemical assay just with a PIK5 kinase, and looking at the amount of ADP that's or ATP that's utilized, essentially looking at kinase activity. And you can see that the potency of this new molecule is very similar to that of apilimod in the low nanomolar range. And consistent with all of those data, what they've shown is that you can administer, when you administer a pilimod orally and you look in tissues, either peripheral or in this case in the brain, these are in neurons, and they look for signs of target engagement, essentially PIK5 inhibition, by looking at the size of those early endosomes. So if PIK5 is being inhibited, those early endosomes don't transition into late endosomes and lysosomes. And what they do is they keep fusing with each other and they just become bigger and bigger. And so the enlargement of early endosomes is a readout of target engagement. And what they see is that with a pilimod, 4 hours after injection or administration, you really don't see any change. Whereas you do see a significant increase when you use the new inhibitor that has better biostability. So what we've done recently is start testing these new compounds against infection of SARS-CoV-2, in this case, the pseudovirus. And in this case, what we've done is we've used a pseudovirus with a mutant spike protein that mimics having already been cleaved by TMPRSS2. And so it can actually enter cells either through the early or late pathway. And so this kind of mimics, I think, what you would normally see in a primary lung cell, which has TMPRSS2. In this case, you can see that, this is the infection rate with the vehicle control. Remdesivir has no effect on this aspect of viral infection. Remember, it's more on the replication aspect. Nifamostat is an inhibitor of TMPRSS2 that, as expected with the mutant spike protein, does not inhibit activity because it's already mimicked cleavage by TMPRSS2, but ASR149 has a strong effect at inhibiting the infection of the virus. So I think this is the first proof of concept that this is likely to be something we want to follow up. And what we're looking forward to now is working with Ya-Wen to actually test this in human iPS-derived type 2 cells, which would probably more closely approximate the balance of cell entry between the early and late pathways. And then we also want to do primary lung cells. And then if that goes well, we'll proceed to doing live virus experiments in hamsters or ferrets to see how that works. And finally, is just a final potential another modality, in case the small molecule runs into issues with, let's say, toxicity after long-term treatment or something like that, we have another option, which is to use antisense oligos to suppress PIK5. These antisense oligos are modified with phosphorothioate linkages, as well as linkages to the sugars that prevent them from being digested by nucleases, but also B, cause them to remain in cells for long periods of time. And it turns out that if you use a constrained ethyl sugar conformation, as you can see here, these types of antisense oligos can be intranasally delivered to the lung at high levels. And so that's another way to inhibit PIK5 in the lung. And we've screened hundreds of PIK5 ASOs to find ones that have extremely high potency at doing that. So that's a second option that we're exploring. So just in conclusion, the key information is that SARS-CoV-2 enters cells through both early and late pathways. PIK5 inhibition can block the late pathway. In collaboration with the Curistem, we've identified a novel PIK5 inhibitor with better stability in vivo and is more suitable for an in vivo candidate than existing inhibitors. And this novel inhibitor successfully blocked SARS-CoV-2 pseudovirus infection in vitro. So I'd just like to thank the people in my lab that are working on this project in this difficult time. A lot of these people have been working on PIK5 for a while and now have been starting to switch over to some CoV-2 experiments. And I'd like to thank Ya-Wen for collaboration in her IPS lung models, and Curistem for sharing the small molecules that they've developed for our experimentation in the lab and our funding from CERM and Harrington Discovery Institute. Thanks.
Great, thank you so much, Dr. Ichida. So I'd like to thank all three of our speakers for such interesting talks. So we'd like to open up the floor for questions. So if anyone has any questions, please just raise your hand and I will click on you and allow you to speak. So it'll be in the order of who raises their hand first. So let's start off here. We have Annie Wu. So let's see. I'm going to allow you to talk. All right. Go ahead and ask your questions. All right. Are you there? You're unmuted, I believe.
Some people are writing in the Q&A as well.
Okay, let's see. Well, let's see. Okay. It's Annie Wu, if you're still muted, but you're allowed to speak. If you would unmute yourself and you can ask your question.
Sounds like it's hard. Ita's now.
Yeah. Yeah, hold on. Let's see here. Let's try Ita. You're our.
Hi.
There you
Ita
go.
Here. Thank you for some exciting and great talks. So I have a question. What is for Justin really? What's the relative contribution of this early pathway and the late pathway, or is it different in different tissues?
Yeah, that's a key question, a great question that I think we are trying to find the answer for. So I think it depends on two things: the amount of TMPRSS2 expression on that given cell, and also the amount of cathepsin B or L levels in that same cell. Those are the two key enzymes needed for the early or late. So I think it's unclear.
And also I read that once a cell start expressing the S protein, it starts fusing, it starts helping fusions between the cells and new virus,
yeah,
and syncytia and so forth. So that's also going on. And do you need the tempers too for that? Or
no, you don't need
your PIKFYVE?
Yeah. My understanding is that furin protease enables that cell-cell direct delivery. So that's another aspect. So you can see that there are like these three modes of cellular spread, essentially: early pathway, late pathway, and cell to cell. What I really think it's going to come down to is a cocktail of inhibitors that block these three pathways. I think hopefully blocking each individual pathway has some clinical effect and we can get it approved. And then we can start doing things where we combine them to really stem the spread more effectively.
All right, so we are gonna let's try Anna Wu again. So I'm gonna allow you to talk and go ahead and unmute your microphone and you should be able to speak. Okay, so that one's still not working. So we will we'll move on to the next question. So we have Evelyn Tran. I'm gonna allow you to speak and go ahead and unmute your microphone.
Hi, can you hear me?
Yes.
Okay, great. Great talks, everyone. One thing that has come to my mind across all three of your talks is the mention of using animal models to infect with the SARS-CoV-2. I was just thinking about, the types of species that we see reported to have COVID-2, they reported a tiger, maybe a couple dogs, humans, of course. And I wonder how that information translates into how we do research. Because someone had mentioned using like ferret and hamster models, and obviously we have mouse models and possibly rat. How does that inform us about the tropism of this particular virus? Is there something different about how you use the CoV-2 virus that allows you to use these normal animal models in research? Or can someone clarify that for me just a little bit more? Thank you. Open to all three speakers.
Can I say something here? I think there are many reports for all different kinds of, you have even snakes can be infected by COVID-19 SARS-CoV-2 virus. And I can say a little bit more from the mouse model point of view. As you probably know that there are two major strains. One is C57 Black 6, the other one is bulb C. C57 Black 6 is almost resistant to SARS-CoV-2 virus infection, whereas I know bulb C can be infected by SARS virus, probably also can be infected by SARS-CoV-2 virus. So it's C57 Black 6 mice is more resistant. It has a more stronger Th1 type of response, so it's harder to infect. Whereas bulb C is more Th2 friendly mice, so it can be more susceptible to. It's not just to SARS virus; it's more susceptible to many different other viruses as well. Does that help a little bit?
Yeah, no, it actually generates more questions. Then I wonder, is it then worth studying some of these species that are resistant to being infected? You had mentioned, I guess, a pathway. So is that a potential therapeutic avenue? What if we can activate that in people? Would they be then more resistant to infections in general, not just COVID?
It's more about the at least I know I'm pretty sure a lot of people are aware that the C57 Black 6 people do a human ACE2 transgenic to make it susceptible, because the affinity of the mouse ACE2 just not too low for the spike protein. So people just make a transgenic, or some people make a knocking mice. So it's the same principle to make the human ACE2 in the mice, so that make it susceptible.
One interesting, emerging finding that's been coming out with a low number of studies is that cats can be infected by the virus and they replicate it. It has, the titer is high in the serum, but they don't develop symptoms. So they might have some way of somehow being resistant to the, some of the effects of maybe the immune effects. I'm not sure, but that seems like something interesting as a possibility.
Well, the virus haven't figured out a way to suppress the innate immunity there yet. So yeah. It's all co-evolution. So the virus evolve, find different ways to suppress the host's immunity. If it's successful, it will move forward to do a productive infection.
All right, great. We have a few more questions. We have Brandon Butler. I'm going to allow you to talk. And please unmute your microphone. There we go.
Can you hear me?
Yes.
Okay. I think my question also has to do with like the previous person's question relating to like, well, my question is more specifically as like figuring out the mechanisms for why people are asymptomatic as opposed to symptomatic. And I wonder if there's like any differences in like in the genetics of those who are asymptomatic, and how that could probably compare to like probably what Justin was talking about with cats showing no symptoms.
Well, Justin, you can go ahead.
Go ahead. Yeah, go ahead.
I think the for asymptomatic or symptomatic or severe, I'm sure you know that there are commodity issues. There are a lot of people with coexisting situations.
And there are some interesting differences between levels of ACE2 and potentially some of these other proteases in men and women, or very young people versus older people. So I think that has.
Oh, can I say something? There are sorry. There are interesting report about ACE2 is an X-linked gene, so you can read a lot more about the level of ACE2, the sex linkage of this ACE2 expression level, and people even just injected the human ACE2 to treat the disease, because it will bind the virus spike protein. Just remove it away, but the sex linkage, the X chromosome linkages plays a major role actually. So there are interesting papers on that. So I would just suggest you search for that. If you want, you can email me. I can send you the paper. It's very interesting actually.
Okay, great. So we have a question from Andrea Kovacs. So let's see. If you could unmute your mic.
Thank you. That was all three great talks. My question is about the endosomes that you spoke about. How long can those endosomes remain in the cytoplasm? And is there a possibility that these endosomes could be a reservoir for long-term, like HIV, sitting there in the cytoplasm without being broken down and fusing with the lysosome?
Yeah, I mean, it's really not 100% clear, but they can remain there for quite a long time. In other systems, they can remain there for many hours or days. I don't know how long they can stay there with viral particles inside, but I think somehow it's good enough, it seems, to because this other study that I mentioned, they blocked that pathway with live virus, and they saw like a great reduction. So I think it's enough to drastically slow down their infection or their spread. Whether it's long enough to allow the viral particles to somehow degenerate or die, I'm not sure. But it can be for many hours or a low number of days in other people's systems, as far as looking at these length of endosomes.
But what about some us being kind of hiding out, like in macrophages, and just kind of not doing anything but just sitting there?
Yeah.
Is that or is there a mechanism which by that these endosomes are broken down just naturally and destroyed?
Yeah. There are definitely pathways for breaking down the endosomes and recycling the lipids. I don't know. I think it's unclear what's going to happen, how that's going to affect the viral particles that are in those endosomes. Are they going to be released at that point? Yeah, it's a fair question. And I think that's why you have to sort of move to these in vivo models to really know how it's going to be effective clinically. And we're trying to find the best model. I don't know what it is right now, unfortunately.
Thank you.
So we have, does anyone else have any questions?
Can I ask Ya-Wen a quick question, actually?
Sure.
For your antibody approach to block the TMPAR function, what do you expect, how it would compare to inhibitor treatment? I mean, there are inhibitors already out there.
Yeah, I'm not sure right now. Yeah, so we're gonna go back to do some pilot study. But we're trying to use the monoclonal antibody because first it's more specific, right? So you get less off-target effect from the inhibitor. And like Justin also said that the two protease inhibitor that kind of people use, like it's a broad spectrum serum protease inhibitor. And one of them has very short life, half-life in the body system. The body will kind of clean it within 1 or 2 days. Right. And the other one has like a not very high specificity affinity towards TMP-ISS2.
Yeah.
So we are hoping that the antibodies can kind of overcome this difficulty. And another thing is that the antibody is likely to stay in your system longer.
Yeah, and also there are people saying that the single chain antibody can be picked up more efficiently from the mucosal surfaces. So it's probably also an advantage for you to do this. Yeah.
And we can actually go both ways, go into the system, or we can kind of nebulize it and go into the lung directly.
Right. Okay, thanks.
So we had a couple of questions that came in through the Q&A feature, so I'll just read those. So there's one for you, Justin, and they're asking, do you expect some negative effect from blocking lysosomal pathway in cells?
Yeah, we do. I think it's interesting. There are actually humans that are haplodeficient for PIKFYVE. So they have one copy which has a loss of function, and they're healthy. So I think you can at least inhibit 50% of PIKFYVE activity and be safe. The question is whether that's enough for inhibiting the viral spread significantly. I think it remains to be seen. I'm sure it's not great to fully block lysosomal activity for long periods of time. So we're thinking of like either transient, inhibition or partial inhibition. Yeah.
And then we have another question here, and I'm not sure who it's directed at, but it says, what about the immune response in mice? Many of the effects of the virus are related to that.
You mean I'm trying to answer this question. The immune response in the mouse is in terms of the similarity to human system, or what was the question addressed?
It wasn't, I'm just reading it out there. I'm not sure if they're still on the seminar anymore. That's just what their question was about the response in mice, immune response.
Maybe I think Zia might have been asking, how similar is the immune response to the one that you see in patients, or,
Well, we have opinions about the mouse modeling. But I think to a certain extent, that's what I can see, that they can see the pathology similar. The pathogenesis, the overall process is similar. In terms of the timing, I would really highly doubt that the timing is very similar, because it's so different organism in terms of timing. It's very different. 5 days in a human might mean 2 hours in mice. I don't know. So it's that's what I guess. Timing is very different.
All right. And we have one last question. So this is about vaccines, actually. So based on all these data, how realistic is the timeline for vaccine development for human use?
It's not something.
Ya-Wen, what do you think about that?
I think we're at least 1 year away.
Yeah, I would agree, probably.
Early, Dr. Fauci said it's early March something, I think. Is that what he said? Sometime next March.
Mm-hmm.
Yeah. I would agree that around that time frame. Yeah.
Okay. Well, it looks like we got all the questions answered. So I just wanted to again thank our speakers, Dr. Yuan, Dr. Chen, and Dr. Ichida. Thank you so much for joining us today. And we also wanted to thank everyone for attending. And let's see, we hope that you can all attend our next seminar, which will be June 12th. So thanks everyone. Have a great weekend. And we look forward to seeing you all at our next seminar series.
Thank you. Thank you, Melissa. Thank you, Mary. Thank you all.
Thanks.
Thank you, guys.
Bye-bye.